The electron transport chain, or ETC, is a critical biochemical pathway in cellular respiration. This essential process occurs in the inner mitochondrial membrane and involves a series of redox reactions. The ETC plays a crucial role in converting energy from food molecules into ATP, which serves as the cell's energy currency. The electron transport chain represents the final stage of cellular respiration, where the most ATP is generated, approximately 32 to 34 molecules per glucose. In summary, the electron transport chain is a critical process that drives cellular energy production through redox reactions in the inner mitochondrial membrane, generating most of the cell's ATP supply. The electron transport chain is located in specific membranes within cells. In eukaryotic cells, the electron transport chain is located in the inner mitochondrial membrane. The inner membrane is highly folded into structures called cristae, which dramatically increase its surface area. This is where the electron transport chain complexes are embedded, spanning from the inner membrane into the matrix. In photosynthetic organisms, a similar electron transport chain exists in the thylakoid membrane of chloroplasts. The specific membrane location is crucial, as it allows for the establishment of a proton gradient across the membrane. As electrons move through the transport chain, protons are pumped across the membrane, creating a concentration gradient that powers ATP synthesis. In summary, the electron transport chain is strategically located in membranes that can maintain separation between compartments, allowing for the energy-storing proton gradient to form. The electron transport chain, or ETC, consists of 4 major protein complexes, 2 mobile electron carriers, and ATP synthase. Complex NADH dehydrogenase accepts electrons from NADH and pumps 4 protons across the membrane. Complex 2, succinate dehydrogenase, accepts electrons from FH2 but does not pump protons. Coenzyme Q, or OQ, is a mobile electron carrier that shuttles electrons from complexes 1 and 2 to complex 3. Complex 3, the cytochrome BC1 complex, receives these electrons and pumps 4 protons across the membrane. Cytochrome C is another mobile electron carrier that transfers electrons from complex 3 to complex 4. Complex 4, cytochrome C oxidase, passes electrons to oxygen, forming water, and pumps 2 additional protons. Finally, ATP synthase, sometimes called complex 5, uses the proton gradient established by the electron transport chain to synthesize ATP through oxidative phosphorylation. Together, these components form a sophisticated molecular machinery that converts the energy from electron transfer into the proton gradient necessary for ATP synthesis. Complex 1, also known as NADH dehydrogenase, is the largest protein complex in the electron transport chain. It's a massive L-shaped protein complex embedded in the inner mitochondrial membrane. Complex 1 accepts electrons from NH, converting it to NAD+ in the process. The complex contains flavin mononucleotide, or FMN, which initially accepts the electrons. From FMN, electrons move through a series of iron-sulfur clusters that act as an electron transfer chain. During this electron transfer process, complex 1 harnesses the released energy to pump 4 protons from the mitochondrial matrix into the intermembrane space. This proton pumping contributes significantly to the electrochemical gradient across the inner mitochondrial membrane, which will ultimately drive ATP synthesis. As the largest complex in the electron transport chain, mammalian complex 1 contains approximately 45 subunits and has a molecular weight exceeding 1,000 kilodaltons. Complex 2, also known as succinate dehydrogenase, plays a unique dual role in cellular respiration. It's embedded in the inner mitochondrial membrane between the matrix and intermembrane space. Complex 2 contains several key components: a binding site for succinate, FAD as a cofactor, and iron-sulfur clusters that facilitate electron transfer. What makes complex 2 unique is that it also functions as an enzyme in the Krebs cycle, where it catalyzes the oxidation of succinate to fumarate. During this reaction, electrons from succinate are transferred to FAD, generating FADH2. These electrons then flow through the iron-sulfur clusters and eventually to coenzyme Q. Unlike complex 1, complex 2 does not pump protons across the membrane. This means it contributes to electron transport, but does not directly contribute to the proton gradient that powers ATP synthesis. Complex two's dual role as both an electron transport chain component and a Krebs cycle enzyme makes it an essential link between these two critical metabolic pathways. Coenzyme Q, also known as ubiquinone, is a crucial component of the electron transport chain. It is a lipid-soluble mobile electron carrier located in the inner mitochondrial membrane. In the electron transport chain, coenzyme Q shuttles electrons from complexes 1 and 2 to complex 3. What makes coenzyme Q special is its mobility within the membrane. It can freely move to collect electrons from multiple sources, specifically complex 1 and complex 2. Coenzyme Q exists in 3 different forms during the electron transport process. The fully oxidized form Q is ready to accept electrons. The semiquinone intermediate Q radical contains 1 electron. And the fully reduced form QH2 or ubiquinol contains 2 electrons and 2 protons. These 3 forms cycle continuously as coenzyme Q performs its function of shuttling electrons within the membrane. In summary, coenzyme Q's mobility in the membrane and its ability to cycle between redox states make it an essential link between the initial and final stages of the electron transport chain. Complex 3 is embedded in the inner mitochondrial membrane, separating the matrix from the intermembrane space. This large multi-subunit protein complex, also known as the cytochrome B-C1 complex, is a crucial component of the electron transport chain. Complex 3 contains several key components: cytochrome B with 2 heme groups, a Riska iron-sulfur protein, and cytochrome C1. The complex receives electrons from the reduced form of coenzyme Q, called ubiquinol, and transfers them to cytochrome C. When ubiquinol binds to the QO site of complex 3, it donates its electrons and gets oxidized to ubiquinone. The electrons follow 2 different paths in a process called the Q cycle. 1 electron goes through the iron-sulfur protein to cytochrome C1, and finally to cytochrome C. During this electron transfer, protons are pumped from the matrix to the intermembrane space, contributing to the proton gradient. The Q cycle is a complex mechanism that allows complex 3 to pump 4 protons for every 2 electrons that pass through. It involves recycling of electrons through the B-type cytochromes. In summary, complex 3 efficiently couples electron transfer from ubiquinol to cytochrome C with proton pumping, playing a crucial role in building the proton gradient necessary for ATP synthesis. Cytochrome C is a small, water-soluble protein that plays a crucial role in the electron transport chain. Cytochrome C operates in the intermembrane space of the mitochondria between the inner and outer membranes. It serves as a mobile electron carrier, shuttling electrons from complex 3 to complex 4 in the electron transport chain. Unlike coenzyme Q, which functions within the membrane, cytochrome C is a water-soluble protein that moves freely in the intermembrane space. Cytochrome C receives electrons from complex 3 and delivers them to complex 4, acting as an essential link in the electron transport chain. At the heart of cytochrome C is a heme group containing an iron atom that alternates between reduced ferrous Fe2+ and oxidized ferric Fe3+ states during electron transfer. Unlike coenzyme Q, which operates within the phospholipid bilayer of the membrane, cytochrome C functions exclusively in the intermembrane space. Both are mobile carriers, but they operate in different environments of the mitochondria. Cytochrome C's ability to rapidly shuttle electrons through the intermembrane space makes it an efficient and essential component of cellular respiration. Complex 4, also known as cytochrome C oxidase, is the final protein complex in the electron transport chain. It receives electrons from cytochrome C, a small protein carrier that we previously discussed. The key function of complex 4 is to transfer these electrons to molecular oxygen, which serves as the final electron acceptor in the chain. During this process, complex 4 pumps protons from the mitochondrial matrix across the inner membrane to the intermembrane space. The overall reaction catalyzed by complex 4 involves 4 reduced cytochrome C molecules, 1 oxygen molecule, and 8 protons from the matrix. This reduction of oxygen to water is absolutely critical, as it creates the thermodynamic pull that drives electrons through the entire electron transport chain. Let's review the key steps. Electrons enter from cytochrome C, pass through metal centers within the complex, reduce oxygen to water, and the energy released drives proton pumping across the membrane. ATP synthase, also known as complex VIVE, is the enzyme that produces ATP using the energy from the proton gradient. It consists of two main parts, F0, which is embedded in the inner mitochondrial membrane, and F1, which protrudes into the mitochondrial matrix. A high concentration of protons accumulates in the inner membrane space due to the activity of other complexes in the electron transport chain. As protons flow through the F0 portion down their concentration gradient, they cause the rotation of the central stalk. The F1 portion contains three catalytic sites that cycle through different conformations: loose, tight, and open. As the central stalk rotates, these conformational changes catalyze the synthesis of ATP from ADP and inorganic phosphate. This entire process is driven by the proton motive force, the energy stored in the proton gradient across the inner mitochondrial membrane. In summary, ATP synthase harnesses the energy of the proton gradient to power the mechanical rotation of its components, which drives the chemical synthesis of ATP, the main energy currency of the cell. The proton gradient is essential for ATP synthesis and forms the core of cellular energy production. Complexes 1, 3, and 4 of the electron transport chain pump protons from the mitochondrial matrix into the intermembrane space. This creates a proton gradient with two components. First, a chemical gradient or pH difference. And second, an electrical potential with positive charges in the inner membrane space and negative charges in the matrix. Together, these form an electrochemical gradient across the inner mitochondrial membrane, storing potential energy like a charged battery. This concept is known as the chemiosmotic theory, proposed by Peter Mitchell, who received the Nobel Prize in 1978 for this groundbreaking work. According to Mitchell's theory, the energy stored in this gradient drives ATP synthesis as protons flow back through ATP synthase, much like water flowing through a turbine generates electricity. The electron transport chain carries electrons from high-energy carriers to the final electron acceptor, oxygen. There are two main entry points for electrons into the ETC. The primary pathway begins with NADH. Electrons from NADH enter complex 1, which is also known as NADH dehydrogenase. The electrons then flow to coenzyme Q, a mobile electron carrier in the inner mitochondrial membrane. From coenzyme Q, electrons continue to complex 3, the cytochrome BC1 complex. Next, electrons are transferred to another mobile carrier, cytochrome C. The electrons then flow to complex 4, cytochrome C oxidase. Finally, the electrons reach oxygen, the final electron acceptor, where they combine with protons to form water. There's also an alternative entry point for electrons from FADH2, which enter at complex 2, succinate dehydrogenase. These electrons then flow to coenzyme Q, after which they follow the same path as electrons from NADH. Throughout this process, energy is released gradually in controlled steps. This stepwise energy release allows for efficient energy capture, which is used to pump protons across the inner mitochondrial membrane. The proton pumping mechanism occurs across the inner mitochondrial membrane. Complexes 1, 3, and 4 are key protein complexes embedded in the inner mitochondrial membrane. ATP synthase provides the only pathway for protons to return to the matrix. As electrons move through the electron transport chain, they release energy. This released energy is used by the complexes to pump protons from the mitochondrial matrix into the intermembrane space. Complex 1 pumps 4 protons for each pair of electrons that passes through it. The electrons then continue to complex 3. Complex 3 pumps 4 more protons into the intermembrane space. Finally, the electrons move to complex 4. Complex 4 pumps 2 more protons, completing the proton transport process. This pumping creates a higher concentration of protons in the intermembrane space compared to the matrix, establishing a proton gradient. The inner mitochondrial membrane is impermeable to protons, so they cannot freely diffuse back into the matrix. Instead, protons can only return to the matrix through ATP synthase, which harnesses their energy to produce ATP. As protons flow through ATP synthase, they drive the rotation of its components, which couples the proton gradient to ATP synthesis. Oxidative phosphorylation is how cells produce ATP using the proton gradient established by the electron transport chain. A high concentration of protons builds up in the inner membrane space, while the matrix maintains a lower proton concentration. ATP synthase, a remarkable molecular motor, spans the inner mitochondrial membrane. As protons flow from high to low concentration, they pass through the ATP synthase channel. This proton flow causes the rotation of parts of ATP synthase, which powers the synthesis of ATP from ADP and inorganic phosphate. Oxidative phosphorylation is a highly efficient process. When electrons from NADH enter the electron transport chain, they generate enough energy to produce approximately 2.5 ATP molecules. Electrons from FADH2, which enter the chain at complex 2, bypass the first proton pumping site and therefore yield only about 1.5 ATP molecules. This process, called oxidative phosphorylation, efficiently couples the energy stored in the proton gradient to the synthesis of ATP, the cell's energy currency. Molecular oxygen serves as the final electron acceptor in the electron transport chain. The electron transport chain concludes at complex 4, also known as cytochrome C oxidase. Electrons are transferred from cytochrome C to complex 4 and then to molecular oxygen. At complex 4, oxygen receives 4 electrons and 4 protons, becoming reduced to form 2 water molecules. Without oxygen as the final electron acceptor, the entire electron transport chain would cease to function properly. Electron flow would stop, preventing NADH and FADH2 from being reoxidized. The proton gradient could not be maintained, and ATP production via oxidative phosphorylation would halt. This is why oxygen is absolutely essential for aerobic respiration and efficient energy production in the cell. The electron transport chain is remarkably efficient at converting energy from food into ATP. Starting with one glucose molecule, the cell processes it through glycolysis and the Krebs cycle. These metabolic pathways produce 10 NADH and 2 FADH2 molecules, which serve as electron carriers. These electron carriers feed into the electron transport chain, where their energy is harvested to produce ATP. Each NADH molecule yields approximately 2.5 ATP molecules, for a total of 25 ATP from NADH. Each FADH2 molecule yields approximately 1.5 ATP molecules, giving us 3 ATP from FADH2. Additionally, substrate-level phosphorylation in glycolysis and the Krebs cycle produces 4 ATP molecules directly. This gives us a total yield of 32 ATP molecules from a single glucose molecule. In terms of efficiency, this process captures approximately 38% of the energy available in glucose. This makes the electron transport chain one of the most efficient biochemical processes found in nature. The electron transport chain's activity is tightly regulated to match cellular energy demands. Electrons flow through the complexes, driving proton pumping and ATP production. Several key factors regulate the electron transport chain's activity. First, substrate availability directly affects ETC activity. These include NADH2, ADP, and oxygen. Without adequate NADH or ADH2, electron flow slows down. Similarly, oxygen is required as the final electron acceptor, and ADP must be available for ATP synthesis. Allosteric regulation occurs when molecules bind to specific sites on the complexes, changing their activity. Post-translational modifications like phosphorylation and acetylation can activate or inhibit the complexes. Perhaps the most important regulator is the ATP to ADP ratio, which provides feedback control of electron transport. When ATP levels are low relative to ADP, electron transport increases to produce more ATP. As ATP accumulates, the ETC slows down. These regulatory mechanisms work together to ensure that cellular respiration matches energy demand, conserving resources and maintaining metabolic homeostasis. Different cell types in the body have varying numbers of mitochondria and electron transport chain activity based on their specific energy needs. Highly active cells such as muscle cells, liver cells, and neurons require enormous amounts of energy to function properly. In skeletal muscle cells, mitochondria can occupy up to 40% of the cell volume. These mitochondria have densely packed cristae to maximize the surface area for electron transport chain complexes. Neurons concentrate mitochondria at axon terminals where energy is needed for neurotransmitter release. Their mitochondria produce ATP constantly to maintain membrane potentials essential for neural signaling. Cardiac muscle cells have an exceptionally high mitochondrial content, around 35% of cell volume, to support continuous contractile activity without fatigue. Other cell types have moderate energy requirements and therefore a medium density of mitochondria. Kidney cells rely on mitochondrial ATP production to power the active transport processes necessary for filtering blood and maintaining electrolyte balance. Liver cells have the remarkable ability to adjust their mitochondrial activity based on metabolic demands. Their electron transport chains can ramp up during periods of high metabolic activity. Some cell types have lower energy requirements and consequently fewer mitochondria. Skin cells contain relatively few mitochondria as they can meet most of their energy needs through glycolysis rather than oxidative phosphorylation. Mature red blood cells lack mitochondria completely, having lost them during development to maximize space for hemoglobin. They rely exclusively on glycolysis for their limited energy needs. Perhaps one of the most fascinating examples of specialized electron transport chain function occurs in brown adipose tissue. Brown fat cells contain numerous mitochondria with a special protein called uncoupling protein 1, or UCP1. This protein creates a pathway that allows protons to flow back into the mitochondrial matrix without generating ATP. This uncoupling process converts the energy of the proton gradient directly into heat rather than ATP synthesis. This process, called thermogenesis, is crucial for maintaining body temperature, especially in infants and during cold exposure in adults. Importantly, cells can adapt their mitochondrial content and ETC activity based on changing energy demands. Exercise training increases mitochondrial content in muscle cells, caloric restriction can alter ETC efficiency, and environmental temperature affects brown fat thermogenic activity. Mitochondrial diseases are a group of disorders caused by mutations in genes that encode components of the electron transport chain. These diseases primarily affect tissues with high energy demands, such as the brain, heart, and skeletal muscles. These tissues require significant ATP production to function properly. 3 common mitochondrial diseases include Lee syndrome, a progressive neurological disorder often linked to defects in complexes 1 and 4, MELAS, characterized by encephalomyopathy, lactic acidosis, and stroke-like episodes, and MRF, which causes myoclonic epilepsy and muscle weakness. The symptoms of mitochondrial diseases vary widely, but typically include muscle weakness, exercise intolerance, neurological problems like seizures and ataxia, and cardiac issues like cardiomyopathy. These symptoms all stem from the same root cause. Mutations disrupting the electron transport chain lead to reduced ATP production. The electron transport chain serves as a central hub that connects to multiple metabolic pathways in the cell. Glycolysis in the cytoplasm and the Krebs cycle in the mitochondrial matrix both generate NADH, which delivers electrons to complex 1 of the ETC. Fatty acid oxidation is another major source of both NADH and FADH2, especially during periods of fasting or high energy demand. Amino acid metabolism and ketone body utilization also produce electron carriers for the ETC. These connections show how the ETC serves as a final common pathway for energy extraction. Critically, the ETC regenerates NAD+ and FAD by oxidizing NADH and FADH2. This regeneration is essential for these metabolic pathways to continue functioning. This recycling of electron carriers creates a vital link between the electron transport chain and virtually all energy-generating pathways in the cell, forming an interconnected metabolic network. The electron transport chain is fundamental to cellular energy production, generating most of the ATP in aerobic organisms. Through its coordinated series of redox reactions across the inner mitochondrial membrane, the ETC creates the energy needed for life. The ETC consists of four major protein complexes and ATP synthase, along with mobile electron carriers like coenzyme Q and cytochrome C. Electrons flow through the complexes, transferring energy that pumps protons across the membrane, creating a gradient. This proton gradient powers ATP synthase, which produces ATP, the energy currency of the cell. The ETC efficiently converts energy from food molecules into ATP with an efficiency of about 40%, far higher than many artificial energy conversion systems. Understanding the ETC is essential for comprehending cellular metabolism, mitochondrial function, and many aspects of human health and disease. In summary, the electron transport chain is truly the powerhouse of cellular energy production, connecting metabolism to life's fundamental processes.